The article I studied was on diversity, compositional, and functional differences between gut microbiota of children and adults. The microbiota found in the gut is shown to play an important role in an individual's health. Further information can be concluded by testing how different age ranges or factors can affect the gut health. Different age groups are tested to find differences of the effects of microbiota. 2 separate groups are tested in this experiment. 2,111 children in the age range of 9 to 12, which is labeled Gen R, and 1,427 adults in the age range of 46 to 88, which is labeled RS. Some methods that are used is study population and sample collection. Stool samples are taken from children and adults to test for differences in gut microbiota. DNA isolation. Random samples were taken from a negative 20 degrees Celsius incubator and thawed before isolation. DNA is contracted and then stored separately in another negative 20 degrees Celsius incubator. 16S ribosomal RNA gene sequencing. Machines such as SequelPrep normalization plate kit and Radjabzadeh were used and able to assess pools of samples for DNA extraction. Data preprocessing. More software is used to read each DNA sequence and then sorted by each category. Then a table was set up for microbiota results and was subcategorized into 5 taxonomic levels: phylum, class, order, family, and genus. Some additional methods after testing were that some patients that were tested were given antibiotics to identify the effect of them on the gut bacteria in children and adults. Vancomycin and ansamycin were given to both groups. Vancomycin is a glycopeptide that is against gram-positive bacteria, since gram-negative bacteria is resistant to it. Ansamycin is an antibiotic against gram-positive and gram-negative bacteria. These are the characteristics of the final data sets of the 2 cohorts: Gen R, which are the children, and RS, which are adults. The number of observed taxa at each taxonomy level. The top indicates the number of unique OTUs identified in each taxonomic clade. Top A is the RX cohort, and top B is the Gen R cohort. The bottom donut plots indicate the average relative abundances of the top major phyla in each cohort. To validate the data sets, scientists compared the average phylum level profiles of these cohorts with those of cohorts of other similar characteristics. Some gut microbiota differences between the children and adults were the increased vitamin presence that occurs with older age. Vitamins such as B2, B6, and B9 were found mostly in children. However, B1 and B5 were found more in adults. Scientists say that the increased amount of B1 and B5 in adults play a big role in cell division and folate, which is important for growth of a fetus and baby during pregnancy. The results of antibiotics, which were vancomycin and ansamycin. The vancomycin biosynthesis had a better effect on the children compared to the adults. On the other hand, the ansamycin had a better effect in adults than in the children. Scientists believe this is because there is a difference in antibacterial activities within the gut of children and adults. In conclusion, there were significant differences between the gut microbiomes of children and adults. Some examples are the differences in microbiota diversity and the firmicutes and bacteroidetes abundances shown in the donut graphs. These were caused by effects of functional properties such as metabolism, antibiotic intake/resistance, and the production of specific vitamins. This provides more information about what kind of factors play a role in gut microbiota that reside in different age groups such as lifestyle, health, and disease outcomes.